My name is Chris Mathis, and I'm the Chief Commercial Officer at Metrion Biosciences. At Metrion, we want to know what you're thinking about these days. As a preclinical researcher, what are your research needs and what stage are you in the drug discovery process? Are you at the beginning, at the hit to lead stage? If so, Metrion can help you find quality hits with high-throughput screening against ion channel or GPCR targets. Or are you at the lead optimization stage where timely medicinal chemistry support is critical? Maybe you need 5 to 10 compounds tested per week against ion channel targets in patch clamp assays. The success of the project depends on providing data to the chemists on your team. Metrion can help. I'm joined by Dr. Gary Clark, Director of Screening Technologies, and Dr. Eddie Stevens, Chief Scientific Officer at Metrion. So Gary, for these early drug discovery projects, what can Metrion offer for hit-to-lead phase of preclinical drug discovery?
At Metrion, we've got a variety of different platforms with different capacities and throughputs. So we have at the higher end a Flipper Penta, which is a fluorescence-based instrument, and then we can go down to our automated electrophys equipment where we have a Qube, which is a 384 well device instrument. Mm-hmm. And then we can progress down to our Q-Patch 48 systems, and then go to manual patch clamp systems as well. But in addition to that, we have other platforms as well to support our drug discovery cascades. So we have other platforms such as a Volta, which is used for our cardiac safety. In a high-throughput screen, what's a typical number of compounds that you might screen for? It does vary from client to client, but anywhere from 10,000 compounds to 150,000 compounds. It does depend on the platform. Some platforms are fast. The Flipper, for example, is a quicker platform than our Qube. So we will always advise on what we think is the best platform for the particular ion channel target that we have. And what are the key elements of the high-throughput screen? I imagine the cells and the cell quality is an important part of that. Exactly, that's right, Chris. So, the quality of the cells and getting reliable recordings is absolutely critical for a high-throughput screen. Because we're only testing the compounds at a single concentration once, we need to ensure the assays are completely robust and will give us the most reliable data so we can identify those compounds that are active against our channels. And Metrion has a fairly extensive catalog of ion channel cell lines and assays. Tell us more about that. Exactly. So we, as you say, we've got an extensive cell line catalogue which cover a lot of the ion channel families, including voltage-gated and ligand-gated channels. But in addition to that, we also create our own cell lines, and we have that actively ongoing as an internal project. But also we like to then create new cell lines for clients when they come and ask us. Excellent. How long would that take typically for a typical ion channel cell line? If we're looking for a stable cell line, it can take sort of up to 3 months or so, but there are other techniques that we can use, such as transient expression or using backman systems as well. Do you want to add anything to that, Eddie?
We've had some real success expressing targets that are hard to express. Mm-hmm. So we've got some cell lines that are quite unique. Can you give us some examples of those? Yeah, NAV 1.8, so that's historically been really challenging, and we've got a really robust expressor, which is a fantastic reagent for screening. And is that available on high-throughput screening? It's available on the high-throughput assays, so there's success with Flipper, Qube, and all the way through to manual patch clamp for more mechanistic studies. Moving on to lead optimization, what types of services and offerings does Metrion provide in that area? Using our cell lines that we have, we design assays that can be bespoke for our client, or they can use our off-the-shelf assays. Mm-hmm. So we'll incorporate the targets that we need. So obviously the primary target we'll be screening on a regular basis. Compounds maybe 10 a week or more, or maybe doing it every 2 weeks. And then we can incorporate other selectivity targets in there as well to give that information back to our client. And coupled with that, we can use our cardiac safety panel as well to really add value to the data that we give to our clients. As far as case studies, can you tell us more about the KCNC1 project that we did for a rare epilepsy foundation? We were approached by Polara and the KCNC1 Foundation to help them find a repurposed drug against a particular variant of the KV3.1 channel. We went about, first of all, making cell lines for both the wild-type KV3.1 and also the particular variant.
and we tested those and characterized those cell lines. And it became clear to us that the variant was a gain-of-function variant, and so we decided that we needed to identify inhibitors of that particular variant. We took that onto the flipper because speed was of the essence for this program, and we screened the library from the Broad Institute and obviously found a number of exciting sort of drugs that were available on the market that could be prescribed by clinicians for.
That's a great example of what Metrion can do.
Exactly.
Eddie, you mentioned hard to make ion channel cell lines. Obviously a big one is NaV 1.9. Can you tell us more about that?
Throughout my career, I've been involved in NaV 1.9 or pretty much failure to express NaV 1.9, but we've really made a step forward here where we've got a robust NaV 1.9 cell line that's suitable for Qube screening, so that can really have an impact for identifying novel analgesics.
That's great news for the pain research community.
And we're also adding a rat NaV 1.9 cell line as well, so that's ongoing and making great progress.
It's important for the translational aspect of pain research.
So the key to the project was the really high quality of our assays. So that's key to everything that we do, but also the really extensive expertise in electrophysiology and ion channels at Metrion.
As a CRO, Metrion is quite flexible in the way we work with clients. We can do fee-for-service work, FTE, and strategic partnerships are ways that we connect with clients. Eddie, can you explain how this works?
The key word is flexibility. So at Metrion, an FTE project will be typically 1 to 2 FTEs, and it's a way to get access to a wide range of platforms and really tap into the expertise at Metrion.
It seems like there's a natural evolution in the way clients partner with Metrion, right? They typically will start with a fee-for-service project and then over time evolve into more of a collaboration or research project.
That's right. So you know, I guess that's about the ability to really answer scientific questions, evolving into the FTE model, where Metrion can really add value to customers' programs.
And I guess a strategic partnership would be more of a long-term commitment, and where Metrion is really an extension of the biotech or pharma company's own lab or program.
Yeah, definitely, because of course Metrion has like deep expertise in a whole range of targets, so it's not just using platforms, it's using that expertise as well to help further a project.
That's true. Many of the scientists are actually from pharma and biotech companies. They have real-life drug discovery experience, don't they?
That's right, yeah, no, so across a range of pharma, so AZ, Pfizer, yeah, a whole range.
Are you thinking about cardiac safety throughout these early days of drug discovery? Cardiac safety is an important part of preclinical drug discovery, and Metrion can provide help with off-target ion channel screening and other translational assays. Gary, can you elaborate more on how Metrion can help with preclinical cardiac safety?
Here at Metrion, Chris, we've got a variety of different cardiac safety ion channels that have been validated on all our patch clamp systems. Mm-hmm. They include hERG, NaV 1.5, and also CAV 1.2, plus other channels in the SIPR panel as well. That offers us the ability to look at individual compounds for a client in greater depth, or look at large numbers of compounds and get an indication of any cardiac liability. But in addition to that, we have other platforms where we can look at more phenotypic responses. For example, we have the Volta, which is a high-speed camera for measuring fast changes in fluorescence as a result of changes in the membrane potential. So in essence, it's measuring an action potential via fluorescence.
What if a client has some kind of borderline hERG liability, where it's not a clear hERG blocker and it's definitely doing something? What would you recommend to the client like that for cardiac safety testing?
The Volta is definitely a good tool for that, because that will give you an idea of how that's changing a system where all the ion channels are being expressed. But we can use other systems as well. We have a CardioExcite, which looks at contractility as well, but also manual patch clamp assessment of cardiomyocytes or iPSC, cardio-derived myocytes, is also really powerful.
And that really fits in the entire preclinical phases of drug discovery, where you have early discovery where you're looking just to make sure you don't block hERG, and then later on you kind of fine-tune the process and would focus on manual patch clamp, higher resolution.
Exactly, and at that point as well, if you're looking for an IND filing, we'll do GLP hERG for you as well.
Eddie, can you talk about the track record of Metrion in cardiac safety testing?
Yeah, so we've got a great track record in terms of being part of the CIPA initiative, where we're involved in characterising the cardiac panel. But also, the point is that we're not just looking backwards. So Metrion constantly wants to enable our customers, which is why we adopted the Volta. It helps answer very clear questions about ventricular action potentials.
So Gary, you mentioned GLP hERG testing as part of the IND package. Eddie, other companies provide GLP hERG testing. What sets Metrion apart in this process?
So I think it's our deep expertise. So we have a huge track record in electrophysiology. So we're absolute experts in it. And also our attention to detail. So quality is fundamental to Metrion. Yeah. Okay, and that also leads to the quality of the reports that we generate. So we generate very high quality data and excellent reports.
Yeah, at that phase in the process, companies don't have time to mess around with reports. They need to have something that's ready to be submitted to regulatory agencies.
Yeah, absolutely. The quality is fundamental to us as a company.
So Eddie and Gary, what makes Metrion a leader in ion channel screening, and why should researchers work with us?
I think the key thing for me is the quality of our scientists. They're passionate about ion channels and electrophysiology, but they've also got a huge amount of experience and breadth of knowledge.
It's about trust. We always focus to timelines, and we always deliver quality.
If we haven't got the right equipment or the right tools for the job, that doesn't work either.
So you've got the breadth of capabilities and the depth in scientific expertise. That's really what sets Metrion apart.
Yeah, and also our relationships with customers. We're easy to work with. We're nimble, we're flexible. So we always try to achieve a great working relationship.
Really an extension of their own laboratory.
Yeah, no, we have the wide range of expertise to easily do that.
Thanks, Gary. Thanks, Eddie. Now I'll hand it over to the lab team.
Hi, I'm Thomas Hill, Senior Scientist at Metrion. Here at Metrion, our scientists have decades of experience in undertaking high-throughput screens, often involving more than 500,000 compounds. It is often an expensive, time-consuming process which can return false positive results. What sets Metrion apart is our specific high-throughput screening knowledge and experience across multiple ion channel targets. Our HTS assays are cost and time efficient. They support hit identification, hit to lead optimization, and selectivity profiling. Metrion has an extensive range of ready-to-go ion channel assays. We can also generate custom cell lines, including complex multi-subunit channels to meet the specific requirements of your projects. Metrion scientists have over 30 years experience in molecular biology and stable cell line generation for high-throughput screening and compound profiling. You can trust Metrion to develop robust cell lines, which result in assays which give consistent, reproducible, and biologically relevant data. This can significantly enhance the efficiency, reliability, and success rate of identifying new therapeutic agents. We also use our expertise to innovate, develop new assays, and enhance our current capabilities. One such project was exploring different sealing solutions on the Qube. My colleague, Catherine Hodgson, Senior Scientist at Metrion, will talk about this.
Hi, I'm Catherine Hodgson, Senior Scientist at Metrion. We offer high-quality ion channel screening services using the QPatch 48 and Qube 384 automated patch clamp systems as higher-throughput alternatives to manual patch clamp. Our highly experienced electrophysiologists use these platforms to develop, optimize, and validate a wide range of assays to benefit client programs, from high-throughput screening through to bespoke biophysical or mechanism of action studies. Automated patch clamp often requires the use of seal enhancers to form the high-resistance seals needed for high-quality patch clamp recordings. It is hypothesized that high concentrations of extracellular calcium and intracellular fluoride give rise to calcium fluoride precipitate at the solution interface, fostering seal formation. Fluoride can affect intracellular pathways, altering ion channel function, and is not optimal when recording from calcium-activated channels. So Metrion collaborated with Sophian to determine whether other insoluble salts can act as seal enhancers, or whether this property is unique to calcium fluoride. We found that although there is no correlation between the solubility constants of different salts and seal resistances, barium sulfate is an equivalent seal enhancer to calcium fluoride. Unlike fluoride, which caused a depolarizing shift in the voltage dependence of inactivation of NaV1.5, sulfate did not alter NaV1.5 biophysical properties. Also, NaV1.5 and Cav1.2 pharmacology did not differ between the two seal enhancers, demonstrating that barium sulfate is a viable alternative seal enhancer to calcium fluoride.
Thank you for watching. Hopefully we've got you thinking about your preclinical research and have learned how Metrion can partner with your team.